The overall goal of this procedure is to clarify the entire liver and the junction with the intestine for global structural and cellular analyses of biliary degeneration in metamorphic sea lamprey. This is accomplished by first fixing the tissues in the hydrogel monomer solution. Next, the hydrogel is polymerized with the fixed tissues. Then, the plasma membrane of the fixed tissue is removed and the tissue becomes transparent. Finally, immunofluorescent staining is performed. Ultimately, laser scanning confocal microscopy is used to show global structural and cellular changes during biliary degeneration in metamorphic sea lamprey.
This method can help answer key questions in the biomedical field, such as the connectomes in the central nervous system, and in our case, the biliary degeneration during sea lamprey metamorphosis. The implications of this technique extend toward therapy or diagnosis of human biliary atresia, because this rare congenital infant disease shares similar characteristics with the biliary degeneration during sea lamprey metamorphosis.
We first had the idea for this method when we found a news article at Nature and presented the online video during our lab meeting. Demonstrating the procedure will be Ann Scott, a graduate student from our laboratory.
After making phosphate buffered saline and phosphate buffer, or PB, according to the text protocol, in a fume hood, prepare 400 milliliters of hydrogel by combining distilled water